welcome you to our final webinar in the microbiome farming series, because there's more to come, of course. Today will be focusing on farmers and microbiome farming. I'm delighted to introduce this session. My name is Claudia Costa, and I'm the director of the Food for Sustainability Academy. A short reminder, you probably know Food for Sustainability is a collaborative laboratory located here in Portugal that aims to solve large-scale problems in the food systems to achieve more productivity and climate resilience. And this webinar is brought to you by the GROW initiative, which is part of the EIT Food. GROW is a farmer-centric project aimed at empowering farmers to shape a sustainable and resilient food system. And today, we have three distinguished speakers who will share their expertise on this crucial topic. With us, we have the pleasure to have Noelia Franco. Noelia is a soil scientist at the Technical University of Munich. And Noelia, let me advance my presentation so we can all be tuned. And Noelia will be detailing the role of soil microbiomes in climate resilient agriculture. We also have the pleasure to have with us Cristine Morgan, the Chief Scientific Officer at the Soil Health Institute. And Cristine leads soil health research programs and is also editor-in-chief of two prominent soil science journals. And also, again, we have the pleasure to have with us David Santos. David is a senior project manager at Food for Sustainability, who is going to detail what living labs are and how you can take part in these pilots, pilots dedicated to promoting soil microbiome. Warm welcome to all the speakers here, and they will be for sure providing with all of us valuable insights into how microbiome farming can boost agriculture productivity, reduce water usage, and contribute to a more sustainable future of our food systems. I will also take this opportunity for a very short reminder that Food for Sustainability as a soil health laboratory. And it's by examining the microbial communities present in the soils and, of course, their associated functions, we help farmers gaining valuable information to optimize their agriculture practices. Indeed, our laboratory capabilities extend beyond the simple analysis, and they also include comprehensive soil health assessments, the application or the recommendation of application of microbial inoculants, and continuous monitoring to measure the impacts on crop performance and soil quality. So if you want more information, I'll leave you again our email. You can find it here, arrudalab@foodforsustainability.org. So after this very short introduction, let's do our usual warm-up with some facts about soil microbiology. I believe that Soraya and Sylvia have prepared a few questions for us on Slido. Is this right? Yes, and we can see Slido here. So I invite you all to use either the QR code here. You can use your mobiles, or if you prefer, using your PC, please type in www.slide.com, and in the box, just insert F for S Academy, and let's see what the team has prepared for us. We will dive more about knowledge about the soil microbiome. Sylvia, Soraya, what have you prepared for us? Aha! So, which type of mycorrhizal fungi forms an extensive network, often referred as the wood wide web? I don't know if you have heard, I think this is a brilliant expression, the wood wide web. And we have here four options: arbuscular mycorrhiza, they are very well known, the ectomycorrhiza, ericoid mycorrhiza, and orchid mycorrhiza. Okay? Is everyone voting? See, I think we have our counting stabilized. So our muscular mycorrhizae are, as we all know, the most common type, and they form associations with about 80% of plant species. They penetrate in plant cells, but they not penetrate cell membranes. But the correct answer is indeed ectomycorrhizae. And these fungi, they form a self around the plant roots, and they create an extensive network of fungal threads, which are called IFA, in the soil. And it is this network that it became known as the wood wide web, connecting different plants and facilitating, of course, nutrient and information exchange. Just for your information, ericoid mycorrhiza, these are specialized fungi that associate with plants in the Ericaceae family, which is, for example, the family related to blueberries. And the orchid mycorrhiza, again, very specialized fungi to orchids, and they are critical for seed germination and early growth. So thank you so much. Let's move on. So ectomycorrhiza, they define the wood wide web. And now, let's become more technical. Do you remember what processes, primarily driven by soil microbes, is crucial for converting atmospheric nitrogen into a usable form by the plants? Okay, let's vote. Ammonification, denitrification, nitrification, nitrogen fixation. Okay, they all involve forms of nitrogen, but different forms. Okay, let's just give time to everyone to vote. I think we have our votes. So ammonification refers to the process of converting organic nitrogen to ammonia. Okay, so it's not ammonification. Denitrification is the process that converts nitrates back into atmospheric nitrogen. So it's not denitrification. Nitrification is the biological oxidation of ammonia to nitrate and then to nitrates. So it's not nitrification. So it means that it's nitrogen fixation, which is the process by which atmospheric nitrogen is converted into ammonia and other molecules that are then available to plants. And we know that this is performed by bacteria and archaea because they do a very symbiotic relationship with plants. So nitrogen fixation is the process of converting atmospheric nitrogen into a form that is bioavailable to plants. Very good. We have one more question. Okay, last question. Which management practices can potentially increase soil microbial diversity while simultaneously addressing issues of soil compactation? So we have two functions here: increase microbial diversity and address compactation. So we have controlled traffic farming, precision agriculture, biochar application, or the use of cover crops. Biochar application, precision agriculture. So we have use of deep root cover crops at the top. Let's wait for everyone to vote. We had 20 of you taking part. So controlled traffic farming doesn't seem to have a lot of preferences. Oh, someone voted in controlled traffic farming, but this practice restricts heavy machinery and is specific mainly to avoid the overall soil compactation. So, but it doesn't have direct effects on enhancing microbial diversity. So it's not controlled traffic farming. Precision agriculture, we know that it's the use of technology to optimize crop inputs, but this doesn't relate straight to soil compactation or has a direct influence on microbial diversity. Biochar application While biochar improves soil structure and supports microbial life, we cannot establish a direct relationship with the issues of soil compactation. So the correct answer is the use of deep-rooted cover crops. And we are talking about crops such as radish or chicory that can break up compacted soil. So compactation on one hand and has these crops grow and die, they add organic matter to the soil, feeding diverse microbial communities at different soil depths. So, use of deep-rooted cover plants was our answer, and it comes to an end to our warm-up. Thank you so much for everyone that took part. Thank you so much, Sylvia and Soraya, for your assistance here. So now let's turn to our speakers. So while Noelia comes to the stage, let me properly introduce you to Noelia. She's a soil scientist at the Technical University of Munich with a deep interest in increasing long-term carbon sequestration in soil through novel agriculture land management practices within the context of climate change. She has developed strong expertise in fields such as physical chemistry, agronomy, and climate change. She's also passionate about communicating science. She coordinates and leads various projects focused on soil and water conservation and their different sustainable management practices. Noelia has also presented her work at numerous international conferences and symposiums, establishing a network that includes soil scientists, farmers, politicians, and non-governmental organizations. Noelia, it's a pleasure to have you here, and the floor is now yours.
Thank you a lot, Claudia. I don't know if all of you can see my screen. If you can confirm
Yes,
this.
we can see,
Okay.
but not in presentation mode.
Okay, because I can see in presentation mode now. Still not?
No, we see the presentation with the slides on our left hand.
Okay, I will try again. Now, you can see?
You're not sharing, Noelia. You stop.
I am not. Okay, sorry. I will set in again.
We can go with your presentation without presentation mode, if easier.
Now?
No.
Okay,
Do you have two screens?
No, I only have one.
Okay.
But it's fine. We can follow your presentation.
Okay,
but you need to click.
I will click and you can see. So yeah, thank you, Claudia, for inviting me to be with all of you today, and good afternoon, so everyone, and thank you for the presentation. So for before to start to talk about microbiome, I would like to start to talking about soil. And I would like to not give you a really complicated definition, but I would like to that you can understand in a simple way why we need to start with the soil, because the soil is mainly the skin of the hair, and in the soil occur the most important biogeochemical activities and processes in the hair crust. In fact, the soil is a non-renewable resource, and currently 60% of the soil related with the ecosystem in Europe are degraded. A simple way to see the
Noelia,
soil.
sorry to interrupt. Noelia, you're not moving the slides.
I am not moving that, so I don't know.
Yeah, now it's fine. Thank you.
Okay, so then I will be like this. You can see now.
Yes, we can see the slide.
Yeah, so as I was say before, so a good way is to use the soil as a mirror. If we have healthy soil, then we can say that we have a healthy society. And if we have degraded soil, this means that the society is quite sick. Um, in fact, the soil is an interface between several different spheres, the atmosphere, the biosphere, the hydrosphere, and the lithosphere. And between all of these spheres happen the more important biogeochemical processes. Today, we will focus in the soil microbiome, because they have an important role in the different our ecosystem. And this soil microbiome is affected by two main controlling factors. Can be biotic factor and can be also abiotic factor. As abiotic, we have plant species, microbial predator, competitor, and as abiotic, we have different physicochemical parameters of the soil, and they can affect to the soil microbiome in a temporally or in a spatial way. But why the soil microbiome start to increase regarding to the knowledge over the past decade? This is important because the soil microbiome have a really important and key role in the ecosystem health in the different agroecosystem, and also currently under a special climate change context. So there is solid scientific evidence that demonstrate a strong connection between different crop production practices and the soil microbiome and the combined effect of green gas fluxes that can be, for example, CO2 emission, nitrogen, the methanogenic processes. For example, here I use a slide that describes really well the carbon cycle, and you can observe how the different microorganisms take part in all the steps of this cycle. But how the agricultural management practices affect the microbiome? The agricultural management practices affect the microbiome because alter the soil. And these changes can affect in a negative way or also in a positive way, and can affect also the diversity, the composition, and the abundance of the microorganisms. I recollect several management practices, and I split the different management practices in 10 group, but there are a lot, and there are even more. And also, the most important that I would like that you take at home is that there are a lot of studies regarding two different agricultural management practices, but they only studied one management practices. But the reality is not this. The reality is that always there are a combination of several management practices. There are not only one. So this is that why we need now focus even more in the combination and how the combination of these different management practices affected the different soil function and subsequent the different soil microbiome. But yeah, if some of you know different management practices, I will be so happy to talk with you and discuss and also learn together. Starting with land use changes, we need to pay attention that the different land use changes, for example, if you have a forest and now you, instead to be a forest, you want that this be an agriculture soil, these 100% affect the soil microbial population and also affect the function of this microbial population. We have to pay attention a lot in the legacy effect, because if now we have a agriculture soil, and in the past this was, for example, a forest or a grassland, of course we will have a legacy effect from the previous microorganisms that were there. In one of my studies in 2015, we found a legacy effect after 22 years of the afforestation of a degraded area. This was a forestation combined with organic amendment combination. And we observed that the quality and the quantity changes in different organic carbon fraction increase the agaromycotina group. This is a fungal group for the Basidiomycetes. And they produce a considerable amount of mycelium that help to the formation of the aggregate. In addition, we found other group of fungi, the ectomycorrhizal, that they facilitate the nutrient uptake by the host plant, that there was Rosmarinus officinalis combined with Pinus alepensis. So in conclusion, they promote the physicochemical stabilization of the organic carbon in different aggregate in a long term. So this makes the soil structure of this soil very resilient. Now we move to irrigation, and there are some studies that demonstrate that sometimes the irrigation have a really high impact. Sometimes it's even higher than, for example, the nitrogen fertilization. And usually the irrigation have a stronger effect in the topsoil than in the subsoil. But in almost all the studies that are until now, they studied, for example, the amount of water, or if there is drip irrigation or is a flood irrigation, but some of them, a lot of them, they don't take into account the quality of the water. And this is something that you need to also be careful, because they can change a lot the soil microbiome. And this means that the soil function will change, of course, a lot. Because, for example, if the water doesn't have a good quality, can introduce several bacterial or heavy metal that can make resistant genes. And then they can modify the ecosystem a lot. Regarding to the tillage, I group the tillage in conventional tillage and in conservational tillage. As conventional tillage, I would like that you understand this kind of tillage, that they use a heavy machinery, and at the same time is doing several times per year in a really deep way. What happened with the conventional tillage, that because this tillage homogenize the soil a lot, make the habitat for the microorganism less diverse. Also, they reduce the soil organic matter, destroy the soil structure, and the study mainly demonstrate that there are more changes in fungi communities due to the destruction of the I5 and the connection with this I5 with the root system that in soil bacteria. As a conventional tillage, we have the no-till, that several studies demonstrate that increase the diversity, the richness, and sometimes also the enzymatic activities. But the thing of this study is that the changes are acute mainly in the topsoil. But with the reduced tillage, the several studies, not only in semi-arid areas, also in temperate areas, they demonstrate that this reduced tillage increase the microbial community and also accelerate the formation and the protection of the new organic matter into aggregate. Why? Because this reduced tillage put in contact the small soil particle together with the microorganism, and this helped to accelerate this protection. And we can find the very good and positive effect, not only in long term, also in really short term. As a crop diversification, I put here plant diversity, crop rotation, cover crop, and crop residues. Of course, always when we cover the soil, there is a reduction of erosion and nutrient leaching. Yeah, they positive contribute to microbial abundant activity, diversity, but also the cover crop maintain the soil aggregation and the porosity. This mean a more resilient soil structure. And there are studies that also found that the cover crop together with the crop rotation improve the soil microbiome, and at the same time, the soil microbiome help to other organisms than the earthworm community with this positive effect. With the diversification of different crop, we can also find more diversity in the different habitat, especially if we include legumes in this crop rotation. And yeah, of course, the plant introduce into the soil a lot of compounds through the root litter and also the root secretion that they also have positive effects. Sometimes there are studies that they don't find effect, but because this study was also not in a long term. But yeah, in these soil microorganisms. Then as fertilizer, we have organic and inorganic fertilizer. They can have a positive effect into the soil, but of course, this will depend a lot of the period, dose, and type of organic fertilizer. But we have to be careful because sometimes this fertilizer can introduce exogenous microorganism into the soil, can also introduce heavy metal that then we can have an increment of the toxicity and reduce the soil microbiome. We have also pesticides and herbicides that mainly they are used in conventional agriculture management practices. Yeah, sometimes and they can also introduce antimicrobial resistant genes into the soil. Yeah, sometimes the pesticides can be longer in the soil after the application. So yeah, so we need to know really well all the soil microbiome and also how is this soil microbiome in the different soil function, to be sure that with the use of pesticides and herbicides, we are not favoring some microbes respect other. The biocontrol is something that's sometimes hell and sometimes even better that use pesticides and herbicides because they avoid the use of chemical in the soil, and they create favorable state for other soil microbiomes. For example, fungi, trichoderma. This is one of the most good example, and improve the soil resistance against abiotic stress. This is something that nowadays is there are a lot of experiments about this because due to the climate change, every time we have more drought, longer drought periods. And we can also have problem with the nutrients. And the microplastic can also create different microhabits. for different soil microorganisms. So there are not a lot of studies about this, but there start to be a lot. Yeah, we need to see what happens in a long term. We have also direct microbial inoculation, but this can be also some kind of risk because sometimes we can favor the microbial inoculants regarding to the native microbial community. So yeah, this kind of biostimulant that promotes the fast and also promote the plant growth. But yeah, we need to know really well the function of the soil microbiome and the different communities of the microorganisms that take part in the different soil processes. Yeah, like other I use here, but I am sure you know also other example. But for example, can be studied where they don't use fertilizer in grassland, and then they found that the fungal community respond really positive regarding to the carbon input that go into the soil. But in other studies, we found that here in the German Alps, the climate change and the different management intensities alter the distribution of phosphor mineralization bacteria and also have an effect in the different arbuscular mycorrhizal fungi in this alpine grassland soil. And yeah, so in my almost my last slide, I would like that all of you pay attention in several points that the soil microbial activities are really complex. So it's important really studied, not only in the taxonomic study, also in the different function that have this microorganism. We also have long-term program, because only with the long-term program we can increase the soil microbial resilience, because sometimes with really short-term studies we have some kind of conclusion, but then because the microorganism community change a lot, then can be really, really different. And yeah, we have to take account the last use history, the legacy, the site-specific condition, because will be not the same if we have a different soil type, if we have different climate condition, and also if we have different cultural management practices. These have to be really considered also. And yeah, one of the ideas that I like regarding to my experience is that this example that Attar and Blaut show us, and it's also something that Fire proposed, is maybe try to understand better the role of microorganism of soil microbiome if we also understand the soil processes and function. So for example, group the different microorganisms according to the different processes function. In fact, this author found that we have in the same soil type, under the same management practices, we found different habitat for the soil microbiomes. This means different soil carbon sequestration. In the macro aggregate that they become after that they were created after the addiction of the labile compound, in this macro aggregate we found more strategist microorganisms, but in the micro aggregate that there was something that was already there in the soil, that was not new create. We found more resistant organic matter in the term of biochemistry thin and also protected in the physical way, and this was more strategist. But yeah, so the important thing here is that the soil microbia have a really important role in the agroecosystem. We need to consider then in the different studies regarding about different combination of management practices, because we need really good agricultural policies. And without this kind of a study, we will never advance in this kind of thing. And also because we need advance in the knowledge of this, because we need model, and we need model that include the soil microbiome. I know this is something so difficult, but we need included this this kind of microorganism. This means that we need we can call that we need a climate smart agriculture, because the climate smart agriculture has three most accepted pillars. They are the adaptive capacity, the mitigation, and the productivity, and all of them are always related with the soil microbiome, because we need efficient execution of the different management practices, and the ecological service can improve not only the carbon sequestration, also controlling the greenhouse gases emission of other compounds of the soil. Yeah, so this is all. I would like to say you thank you for your attention, and I would like to share with you this link here. This is a book that I wrote some years ago together with different colleagues, because we want to explain also the children how important is the soil biodiversity. This is free. You can download this free, and there is in 8 different languages. So yeah, thank you, and sorry a lot for this problem with the screen.
No worries. Thank you so much, Noelia. And I will start with this picture. I have to say, this are very beautiful books for children. The illustrations are wonderful. I only read one in Portugal. In Portuguese, it was really very interesting. Well done. Thanks for also start promoting education amongst the youngest. Noelia, while our audience prepares, most likely they will have some clarification questions. Could you just detail, you mentioned a lot of studies, a lot of practices. Where do these studies take place? Are they applicable to more Mediterranean context, North European context? So detailing a little bit more about where these studies took place. And also, I have something that crossed my mind. We always talk about the benefits for the soil microbiome, and you mentioned some of the management practices that we could use. Is there any possibility that we might be propagating, harmful microbial communities by increasing, the good practices that promote soil health? So, for example, promoting pathogens. Should we do kind of a baseline assessment to really understand what we are promoting, or this is not the case? The ecosystem will always tend to the equilibrium.
Yeah, so the first question is that all of these studies are doing around the world, not only in semi-arid areas. They are also doing in temperate areas, and they are doing in mountain areas with totally different climate. And also some of these studies I also found in volcanic soil. Yeah, so the thing is, as we can talk about a special baseline. Of course, every soil is different, and also will depend a lot about the crop that you have it. and also the cultural situation. But the most important is that we always have the soil protected in a way that we have a really good amount of organic matter, because if we have really good amount of organic matter going into the soil, of course we will improve the I don't know how to call the good microorganisms that they are there. But when I talk about have a really good level of organic matter in the soil, doesn't mean take organic matter from some place and put there. It's not like, okay, I take this amount of manure and I put there. No, it's incorporate this organic matter, and it's to see how this organic matter have an effect in the different function in a long term. I think, okay, there are evolution, and the microorganism can change a lot, and they can develop, and there are successional processes. I am no expert in microorganisms, but now with the climate change and with the different condition of reduced precipitation or increased temperature, there is something that we don't know.
Mhm. So it's again, we need, as you were pointing, some more modules to feed the data so that we can have, you know, this understanding of how things are changing.
Yes.
One other question, Noelia. So based on the research and your own research, what do you think is the most effective agriculture practices for promoting beneficial soil microbial communities, for example, that enhance carbon storage from the list you kindly provide us?
Yeah, for example, in my case, and the best management practices in my study when I work only with agriculture areas was the reduced tillage. The reduced tillage combined with a green manure. The reduced tillage combined with a green manure helped me to protect the organic matter in a deeper layer, but without a really high soil perturbation. And also with this green manure that incorporate legumes, I also use less inorganic or organic fertilizer. So the basal respiration was really good. The enzymatic activities were the best in these management practices compared with other, and also the carbon sequestration in a long term and the resilience of all the ecosystem, also in the soil structure and also in the productivity of this. In this case, was almond crop was really good. Was working really good.
Okay, very interesting. So reduced tillage and green manure. So if you want to know more and continue this conversation with Noelia, please now use the chat. I need to move on. I can see that Soraya has already placed here the link to your books. So I wish happy readings to everyone. So thank you so much. We need to move on. And now, thank you, Noelia. I hope you can stay a little bit longer. And now I will call to the floor Cristine Morgan. So, Cristine, if you want to start sharing your presentation, I'll detail a little bit more about your background. So, Cristine Morgan serves as the Chief Scientific Officer at the Soil Health Institute, where she established research priorities to advance soil health and develops the scientific direction, strategy, and implementation for soil health research programs. Cristine is an adjunct professor of soil science at Technical University, where she tenure and her research emphasizes soil hydrology, pedometrics, and global soil security. Cristine is also an editor-in-chief at Geotherma, which is a global soil science journal, and the founding editor-in-chief of Soil Security. Cristine has served on the board of directors of the Soil Science Society of America and is also a fellow of the Soil Science Society of America. She has earned a master and PhD in soil science from the University of Wisconsin, and their bachelor degree in plant and environmental soils sciences from Texas University. Cristine, I know it's early in the morning. Thanks for being here. We can see your presentation. The floor is now yours.
Thank you very much for that introduction, and hello, everyone. Greetings from North Carolina. It's an honor to be here to get to talk to you all today about soil health. Just to give kind of a level set, I know that this group is very interested in microbes and the soil microbiome, and so am I. I think about it from maybe a different perspective, as opposed to measuring the microbes that are there. I think this presentation will be talking more about measuring what the microbes are doing for the soil. So really about the water. I know you all are in a more arid climate than most of the climates that I work in, but having come from Texas, I do appreciate the recognition that water is really important to agriculture, and how our soils cycle water is also very important to agriculture. And so the title of my presentation today is Measurable Benefits of Soil Health. I'd like to provide a definition of soil health, and for me, it's how well soil is functioning, specifically for the ecosystem service that, as maybe you're managing it for. And in the context of agriculture, we're wanting the soil to help us grow a healthy, productive plant so that we can get food from that plant. And one example that I like to show, now this is a row crop agriculture from the Midwestern United States, but this picture I think is very relevant to all of agriculture. And I like it. Oh, you don't see someone said they don't see the presentation. Claudia, do you see it?
Yes, I can see the presentation. It's in full screen, and probably from your friends. Thanks for letting us know, but we can see it. Thank you, Cristine.
Okay. So essentially, this is an example of one soil and one climate with the same cropping system. And the soil is being managed differently on one side of the fence compared to the other side of the fence. On the left-hand side of the fence, the soil is not being tilled. It's in no-tillage agriculture. And I don't know that there is a cover crop grown on this field. And on the right-hand side, the soil is being tilled. And whether you use tillage or no tillage, it's a really good example how we can have the same soil, the same crop, and different management in the same environment, and we have very different soil functioning. In this picture, it has recently rained, and on the right-hand side, the soil that has been tilled, in the background, you can see that there has been some real erosion inside the field. There's certainly surface sealing of the soil surface, which makes the water route across the field as opposed to infiltrate into the soil and to be stored for the crop. And I think this is something that we commonly see when our soil is not functioning well hydrologically. This is, we see the water not going in and being captured. And of course, this is also a very important function of soil during an irrigation event. We don't want the water to run off. We want it to go into the soil. And then we also want it to be available for the plant and not be kept too tightly to the soil particles and be unavailable to the plant. And for me, this functioning of soil, whether we're interested in nutrient cycling or we're interested in water cycling, carbon farming, or all, climate smart agriculture, from my point of view, soil structure is the foundation of soil health. And what I mean by soil structure is it's the soil's ability to aggregate and best capture and move water through the system. But also these aggregates are a result of microbial and plant root activity. It's from the wetting and the drying of the plant. It's from root exudate, the aggregates glued together from root exudates and from microbial activities. And the microbes also like soil structure. The roots and the microbes are working to create this structure because this is also the habitat that they prefer as well, for many reasons, due to aeration, water, and nutrient use. And so one of the things that we've done at the Soil Health Institute, I should have introduced where I work, so I'll do a quick introduction. We are a small not-for-profit institution. We have about 35 soil scientists and soil health educators that work at our institution, and we try to do translation science so that we can make the case for safeguarding and enhancing soil functioning. And so one of the projects we did was included the United States, Mexico, and Canada, where we went into long-term research sites that were replicated and had controls for soil health management practices. And across these 124 sites, they all had 10 or more years of consistent management in the plots. And these plots were uniformly sampled in 2019. And we took over 30 measurements of the soils. And at each of these sites, we had a control and a treatment of either residue retention, rotation diversity, crop diversity, the addition or non-use of cover cropping, the addition of organic nutrients, and or a decrease in tillage. And this decrease in tillage included, traditional tillage and no tillage, but it also included comparisons of traditional tillage and reduced tillage. And what we did at each of these sites is we made all of these measurements, and I don't want to spend our time together today. Yes, we can send the presentation afterwards, but I don't want to spend all the time today talking through each of these measurements. But I do want to recognize for this community that we did a lot of measuring of the microbiome. So here's a lot of community measurements that we made. We measured the carbon, indicators of carbon cycling. We measured indicators of nitrogen cycling, water cycling, and then other kind of traditional fertility measurements. And what we found in this project, the goal was to select an essential suite of measurements that were available to commercial laboratories so that we could think about a way to monitor soil health across the continental North America. And we wanted measurements that worked in both humid conditions, dry climate conditions, warm climate conditions, and colder climate conditions. And the measurements that we landed on as indicators of improved soil health across these 124 sites was organic carbon, potentially mineralizable carbon, aggregate stability, and plant available water. And I want to stop for a minute and talk about what each of these means regarding soil health. I mean, obviously, organic carbon is something that we're all very interested in agriculture. Plants actually don't use carbon as one of their nutrients, right? But carbon is a good indicator that there's active that the microbes have something to eat, that the nutrients are cycling, and that in general that there is food source for soil, because we all know soil is alive. Potentially mineralizable carbon is an indicator that tells us what is the activity of the microbes in that soil. It does not tell us which microbes are there. It doesn't tell us who's alive and who's dead and who's functioning and who's doing what. And you all know those are really, important measurements, but they're also expensive measurements. Potentially mineralizable carbon is just saying this is the activity of the microbes. And what we do, how we take this measurement, if you're not familiar, is you take the soil from the field, a composite soil sample, you air dry it in the laboratory, and then you put it in a container and you add a known amount of water, and then you measure the carbon dioxide that evolves from that soil in a 24-hour period. That's the way we take the measurement. And so it just gives us a 24-hour CO2 burst resulting from re-wetting air-dried soil. Aggregate stability is one of my favorite measurements because it really integrates microbial activity, root activity, and root health, and tells us something about how water is moving through that soil. And aggregate stability, there are a lot of different ways to measure aggregate stability. I decided not to make today a lecture on all the different ways to measure aggregate stability, but I do want to show you one method because it's a method that everybody on this conversation today could use. So I'll talk a little bit about that. And then plant available water. And plant available water we know is really important. And plant available water is the amount of water that soil is holding that is plant available. And so generally we measure it by measuring the water at field capacity and subtracting the water content at wilting point. And I'm going to show you more about that one. And I don't think anyone here can argue that we want more plant available water in our soils. Okay, so what are these indicators of water cycling? We measured a lot of them. You know, I've already spilled the beans and said we were measuring plant available water and aggregate stability, but I just want to just give some overview that we measured bulk density of these soils. And if anyone here has measured bulk density, you know that's a very hard measurement to make. We measured permanent wilting point, which is in this middle picture here where it's a dried ground soil on a little puck on a pressure plate. And we put it at 1500 kilopascal suction. And then we measure the amount of the gravimetric water to soil ratio. That's permanent wilting point. So it's the water that's held in the soil that's not available to plant roots, theoretically. And then we measure field capacity measured on repacked soils and on intact soils. And this is really important, and something very different about our methodology is we took whole soil cores that were intact, saturated them, put them on a tension table at 33 kilopascals of tension, and then measured the amount of water that those pores in the soil were holding. And that's our field capacity, of which we're going to subtract wilting point from. We also measured saturated hydraulic conductivity, which was something that I added to the experiment. And it turned out that this is a really important measurement that we're all interested in, but it's not scalable and it's really hard. And you have to take a lot of measurements for it to be a useful measurement. So I'll just tell you that that's why we didn't use it. So I'm going to just show you some quick results. Each of these plots are what we found in soil measurement when we did a practice. And so for instance, residue retention, so the 0 line is the mean control for the 124 sites that had residue retention as a treatment. We looked at the control compared to the treatment, and these are the measurements. So here's bulk density, permanent wilting point, field capacity of repacked soils, field capacity of intact soils, so they still have their structure, and saturated hydraulic conductivity. And the way to read this graph, if you look up residue retention, you will see here's bulk density, the mean, and a 95% confidence of bulk density. And you can see that bulk density decreased at about 5% when residue was retained to the soil. So that's good, right? It means that structure was forming from residue retention. Permanent wilting point, we did not see a statistical difference. We don't expect to because that's a function of soil texture. Repacked water content measured at field capacity, again, that's a function of soil texture, so we don't really expect it to be different. But it was very exciting to see a difference of intact water content at field capacity. So again, both bulk density and this field capacity intact says that when we retain residue on the soil surface, we improve soil structure. Hence, we are improving the water relationship and more water available to the plant. And you can see saturated hydraulic conductivity increased with residue retention, but the error, because it's a measurement that has a lot of variability to it, the confidence was huge. So yes, it was a beautiful measurement, but statistically we couldn't say much about it. We see the same, and I think this is very interesting to this audience because you work in a lot of vegetable production systems. Organic nutrient sources also decrease bulk density and also increase the water content held intact. Cover cropping had some effect on intact water content, but it wasn't statistically significant. And decreasing tillage also improved water content, but it was not statistically significant. So I just wanted to give you look at some of the data. But most importantly, the other thing that we did is we created a pedotransfer function for plant available water or available water holding capacity. So again, it's field capacity minus wilting point. And we know that that's a function of soil texture. Clayey soils hold more water than sandy soils, silty soils, all that's different. But when we put soil texture into the model, and soil organic carbon concentration. So one of the things that we found in all of the different soil health management treatments, we had an increase in soil organic carbon over time. And this increase in soil organic carbon was a very simple way to predict the increase in available water holding capacity. And we would love to have direct measurements of soil structure, but they're very hard to do. We also love to have direct measurements of available water holding capacity, but they're also very hard to do. So a pedotransfer function gives us the opportunity to predict something that's very difficult to measure with information that is not so difficult to measure. So soil texture and carbon concentration in soil is not that difficult to measure. And so the cool thing about this outcome of this work is that we were able to find some significant increases in water holding capacity of the top 6 inches of soil or the top 30 centimeters of soil associated with increases in organic carbon and soil health management practice change. And so this was very exciting. One of the caveats I want to say, and I think this is a place where we need to do more research, is we had to take out calcareous soils. And I didn't look up soils of Portugal, but I know Portugal is a very dry landscape. And I think I don't know if you have calcium saturated soils, but when there's a lot of calcium carbonate in the soil, we found that all of our soil health indicators did not change as much as when the soils didn't have calcium carbonate in them. And I think that that's kind of an area of really interesting research. I mean, carbon, microbial activity, all the things were not as pronounced improvement when the soils were calcareous. And it was the same with water holding capacity. So that's a bit of a sidebar. There are places where this pedotransfer function is very useful, which is much of the United States that doesn't have calcareous soils, and Canada and Mexico. But I would caution to use this pedotransfer function in a case where you have calcareous soils. So the other thing I said I would talk about is this aggregate stability. So we know that aggregate stability is a great way to quantify the structural integrity of the soil, the microbial activity in the soil. We know aggregate stability is hugely influenced by management, and there's multiple ways to measure it. And to give you an example, these pictures here to the right is a picture of the same soil taken on the same day. And it was right after rainfall, and the soil on the right has a perennial grass growing in it, and the soil on the left is tilled. I mean, we've all that work in agriculture have seen both of these kinds of soils, but this is just the huge impact that aggregate stability has on water in soils, and plant health in soils, and drought resilience in soil. And so the way we like to measure aggregate stability, so in this big experiment I told you about, we measured all the different ways to measure aggregate stability. And this method was just as good as the methods that were tried and true and very traditional. And this method you can measure with your smartphone, and you can download the Slates application for free on both an Android and an iPhone. And what this app is literally you can just tape it to a jar or something, and you measure pea-sized aggregates of soil before and after they slake in water. So for this method, you need dry aggregates in a container, and you measure aggregates at 0 minutes before they're placed in water and then 10 minutes after they're placed in water. And the app actually does the calculation for aggregate stability for your soil. So the way we sample is we collect the upper soil and we just kind of push it through shake a sieve, and we collect these pea-sized aggregates into a container. We take it back to the lab, and the laboratory, this lab is fancy, right? You could imagine you could do this at your kitchen table, but this laboratory is automating the measurement. And essentially what we found in our North American project to evaluate soil health measurements. So let's see, STAB 10 is the aggregate stability we did with the iPhone. And these other aggregate stability methods on the y-axis are just similar other aggregate stability methods. But again, what we found with residue retention, we had significant improvements in aggregate stability. Adding organic nutrients improved aggregate stability by some measurements and not by other. And we think that a lot of these organic nutrients were manure, like raw manure. And I think that they increased the salinity of the soil and made it more dispersive depending on the methodology you use to measure. Cover cropping improved aggregate stability, and decreasing tillage improved aggregate stability. So these are all things that are also shown in that plant available water stuff, is that we know that if we increase the photosynthesis and the microbial activity and decrease the number of times we disturb the soil, that the soils aggregate better. And we've measured this aggregate stability with the iPhone across the United States now, over 1,600 samples on crop fields, not research sites, but like production agricultural fields. And we definitely find a strong signal of these management practices to improve aggregate stability. Okay, so the last thing I wanted just to mention really quick, because I talked about what are some of the measurable, what can we measure about improvements in soil health? And another thing that the Soil Health Institute has done is we have done partial budget analyses of farms that have adopted soil health management practices. And I don't have a lot of time to go through the methodology today, but you can certainly look on our, so this is just a paste from our website. And this is the web address, soilhealthinstitute.org. And if you go to our institute site, you can just work through our work and initiatives, and it will come up with this page. And if you click right here on this tile that says Economics of Soil Health Systems in 30 U.S. Farms, I will share these slides so you all don't have to memorize any of this. You can click on Economics of Soil Health on 30 farms, and you can see some of the work we've done. This is 30 farms. We've done almost 200, but I chose this 30 farms because it had some examples in arid conditions. And so right here in California, you can click on this, and it's a walnut farm, and you can see the partial budget, and you can see the interview from the farmer if you want to listen to what the farmer says about adopting. So this is a walnut farm that's irrigated, and it's the closest thing I could find to Portugal. And this is okay, and you'll have to excuse the acres because, of course, these are for farmers in the US, so everything's in acres and dollars, and I did not take the time to convert it to your currency in hectares. So my sincere apologies. We've had a lot going on in the US lately, as you can imagine, and we've had to deal with a lot of like contingency plans for our small nonprofit. But so I've been distracted. But the cool thing here is this business case for soil health. This is a 193-acre walnut farm. And the soil health management systems is this farmer has incorporated cover crops. And they're also doing a lot of soil nutrient measurements. And they have found a net farm increase of $425 American USD dollars per acre on their farm from adopting cover crops. Oh, yes, thank you. Yeah, 2.5 acres per 1 hectare. Thank you, Herberto. So anyway, the cool thing is about you can download all of this. It's all free and available. But the nice thing, I think, from a farmer and maybe even a crop advisor point of view, is we not only show we show the reduced and the additional expenses from interviewing the farmer about their management practices. So you can imagine if you add a cover crop, you have to pay for it. So $25 more per acre to buy the crop, but we're the same, and even more money for fertilizer and amendments because they're adding this cover crop and they're probably, irrigating. But the benefits of this cover crop, which happens to be a brassica cover crop, is $440 per acre reduction in the need for pesticides. So this Walmart farmer is using the cover crop to help with pest management. They're also saving money in fuel and electricity. They're saving a little bit of money in labor, but not much. And they're saving a lot of money in equipment ownership. And you can go in and read about exactly how they're saving the money and where they're expending more money. So this farmer is benefiting almost $700 an acre with the cover crop, and it's costing them $270 per acre to implement that cover crop for a net benefit of $425. And the other thing you can see we've tried to, for each of these sites, kind of do a summary of what this farmer's doing. So this soil health management system adopted cover crops. You can see the cover crops that they're planting: rye, oat, wheat, barley, triticale, vetch, mustard, radish. The cover crops, we talk about when they're planted and how they're terminated. Roller crimper is how they're terminated. Brassicas in the cover crop mix were successful in eliminating nematodes, so that's the pest issue that they're dealing with. And they showed the compost expense and other additional expenses, and then how they have reduced expenses. So I think it's very detailed, so you can go read it. That's the takeaway. So with that, I just want to thank you all for your time. Thank you very much for the invitation and the opportunity to talk about soil health. You can contact me at cmorgan@soilhealthinstitute.org. And with that, I'll turn it back over for questions in the time that we have remaining.
Thank you so much. Thank you so much, Cristine. And I can see you have done your homework. You can also thank us in Portuguese. Great.
I've been practicing Portuguese. Obrigado.
Oh, very good. Very good.
But I had to add y'all. Y'all is Texan.
Yeah, all right.
Which is where
Okay,
I'm
so we
from.
learned
Okay.
something. Great, great.
Yeah.
It's Cristine, I think we are a little bit over time, so I'm not going to extend too much in the questions. Please refer to the chat because I need to move on. But just
Yes.
to share with you that I feel that the numbers are very, very important. One thing is science, which is also very good, but to see how this translates into the numbers and to the bottom line of farmers, it's really, really critical if we want to promote the change. So Cristine, many thanks for bringing this. I will have to point you now to the chat because I need to move on.
Absolutely.
I didn't want to interrupt you because it was a lovely presentation, but now I will call to the floor David Santos. David, some of you might know, holds a PhD in agriculture and engineering from the School of Agriculture here at the University of Lisbon. And in the last 7 years, David has dedicated his research career to the study of unconventional feedstuff, such as algae and their potential uses in monogastric diets towards increasing the sustainability of pig, poultry, and rabbit production. And it was in this context that David worked in different European laboratories to carry out research, including Poland, Luxembourg, and Spain. And it were these experiences that have yielded several outputs, contributing for his publishing records of over 30 papers in international peer-reviewed journals. David is going to show us a living lab. I don't know if it is Lilas for soils or Lilas for soil, but David will let us know. Thank you so much. The floor is now yours.
Thank you, Claudia, for your lovely introduction. Thank you, everybody, for still sticking around. Bear with me for this presentation in our project Lilas for Soils, where we are working with carbon farming practices and together with farmers to give them new insights into these the potential that these practices have for their activity. So to give you a bit of a context right now into how this project came up. Currently in Europe, as most of you may know, the primary sector is facing a conundrum, right? So on one side, there's a need for reduction of the environmental impact of production. And this has gone so far as there being European governments like the Dutch one, paying farmers to close doors in order to reduce the pollution of local habitats with nutrient leachings that can cause pollution events, such as the ones related with eutrophication. And at the same time, there are farmers under heavy pressure from trade agreements that decrease the competitiveness of their activity and highlights the different requirements they have compared to farmers from outside of Europe. On another hand, Europe needs to become more independent from imports if it aims at having a sustainable primary sector and an industry that does not suffer from price spikes like the ones seen in wheat when the Ukrainian invasion by Russia began. For instance, more than 90% of wheat in Portugal used for food and feed industries in 2021 came from exports in 90%, which is very telling of the stability of industries that rely on this commodity. Furthermore, the world population continues to increase, which will propel the demand for food to new heights. This duality, of course, brings added complexity for European agricultural policy as well. Moral of the story, we need to achieve food production that works with nature to preserve ecosystems whilst maintaining food production levels on the long term. So all of these influencing factors are worsened by climate change, which has been more impactful in the Mediterranean region. In fact, water is ever more scarce and the dry seasons are increasingly demanding for all types of farmers. This contributes to reduced crop yields, animal productivity, and increases the threat levels to soil health. For example, increased erosion, food security, and overall ecosystem resilience. Plus, considering that conventional farming is a major contributor for greenhouse gas emissions, farming is at the epicenter of a major turmoil, both as a causing agent and as a victim per se, at the same time. It is in this context that our project was conceived. The project gathers 24 partners spread in 8 countries with multiple levels of expertise. These partners represent farmers and industry, academia, authorities, civil society, and NGOs. The overall objective of the project and of these stakeholders being to promote carbon farming practices throughout the creation of 5 living labs, one in Portugal and Spain, the only one that is international in this consortium, France, Israel, Italy, and Greece. In these living labs, we will provide demonstration sites where farmers will implement carbon farming practices during the project, allowing the assessment of soil organic carbon in addition to other parameters that reflect soil health as a positive externality, let's say, into the practices that we are implementing. In addition, different monitoring, verification, and reporting technologies will be tested in these demonstration sites to test different ways of measuring these parameters. And it is at this point probably that I should say what is a living lab, right? This is a new term, a more or less new term that is going around right now. And basically a living lab is not a physical lab per se, and it's rather a network that represents different classes of stakeholders all working together towards a common aim. Okay, so in this case, we are connecting all the types of stakeholders that I mentioned, working around carbon farming practices, from farmers to policymakers working on agriculture policy. So the project, as you see, is very big. It has lots of partners, and it also has a very ambitious agenda. So we will promote over 20 co-creation workshops across all living labs, allowing the exchange of experience not only within the living lab itself, so in a very regional level, but also between the different living labs, empowering farmers from Portugal with the experience of farmers from Israel, for example. In addition, we will promote training workshops directed at enhancing skills related to carbon farming practices. The data withdrawn from these demonstration sites will feed recommendations for policy workshops aimed at policymakers and end users alike. The project aims to implement mechanisms of financial support to third parties via open calls directed at MRV technology providers and more farmers that were not included at the time of proposal, reaching a total of 15 MRV technology providers, so monitoring, verification, and reporting, and up to 100 farmers. All of this work will contribute to significantly reduce the CO2 equivalent emissions in the Mediterranean space, the creation of a network with over 125 stakeholders, and the implementation of carbon farming practices in real-life settings in over 71 farmers, and the creation of awareness on the importance of carbon farming to over 65 policymakers. So in our project in particular, at the time when we wrote the proposal, we defined 5 major classes of carbon farming practices that we are considering. But of course, they are not only excluded to this one. So we're looking mainly into peatland management, agroforestry, maintaining soil organic carbon on mineral soils, livestock management, and nutrient management. These different classes have different objectives and effectiveness, retaining soil organic carbon, which is why we consider more than one often throughout our demonstration sites. Also, of course, reflecting the area where we're working. These living labs of our project will be specialized in these different classes, according, of course, to the characteristic of the region, but also of the farmers themselves. And we will not limit carbon farming practices to this list. We are very open to the input of farmers to conceive new carbon farming practices, to suggest other ones that we can include in this list, which is overall the major objective also of the first co-creation workshops that we will do. So as I mentioned earlier, the project wants to create 5 different living labs in 6 different countries, already represented by stakeholders from all the 5 representing classes that I mentioned as well. And the overall aim of these living labs is to work together through this mechanism of co-creation where everybody gets a say into what we're doing and how we should be doing it. So, and we are right now at a stage of the project. The project started in September of last year, where we are defining the framework of living lab activity, and this will start through our Living Lodge events, which I will speak a bit more in the next few minutes. So regarding the open calls themselves, the Living Labs will open these open calls to new farmers and MRV providers in a very near future. The farmers in particular will be selected in an open call scheduled for March of this year. Of course, this does not mean that we will limit potential partners to those being selected in these open calls. In fact, Living Labs are eager to create partnerships with regional authorities, for instance, beyond those already included, to create a diverse environment that ensures the most fruitful knowledge exchange and contributes to the maintenance of the Living Labs beyond the existence of our project Lilies for Soils itself. Because after all, this is a major objective of these sorts of initiatives in Europe. We want to have our Living Labs working independently after project funding, and we aim to do this by creating strong and long-lasting partnerships. The project itself is made of 3 major areas of work with 2 corresponding work packages each. Since September, we have launched the workshop. In the launching workshop, we have been working hard in establishing a framework for how the Living Labs should be structured to engage the stakeholders to be included in this co-creation process. So we already have included people that were not working with us initially. And right now we are transitioning to a more active phase of the project where we are defining the requirements of farmers to be included in the Living Lab, and the methodology also that we will use in sampling and analyzing the soil samples that will be taken throughout the project. In the future, the generated data will be used for the creation of synergies between our project and others in the European Union funded project list, mainly in the Mission Soil program. Furthermore, we will explore the ways that the carbon credit markets can be used to increase revenue streams for farmers and create a means for effective verification of soil organic carbon sequestration. Overall, the work with the demonstration sites themselves will follow a similar structure throughout all the Living Labs. The analysis of soil samples will be centralized, in our case, in our Living Lab in Nibersoil particularly, in our partner Niker from the Basque Country in Spain, who will perform the physical chemical characterization of soils samples also including soil organic carbon, of course, but also doing the microbiome analysis. And first, what we will do is characterize a plot of land that the farmer has between 1 and 5 hectares, more or less, regarding the practices that are being used, previous soil analysis that they have that can complement the way that we approach our work, and the different objectives of the farmers that the farmers have in place. We will then follow a protocol of sample collection, which will be overseen by living lab leaders, and which will take place every 2 years until 2029. We will then perform different measurements, as I already mentioned, but also looking not only in one single indicator, but also looking into positive externalities that we identify in the farm, in the plot of land as a whole. So now I will transition from speaking about Lilith for Soils itself to speaking about our living lab in particular, Ibersoil, Iberian living soils. So I'm going to narrow down a bit more into the structure of our Portuguese and Spanish living lab. So the consortium of the living lab itself is led by Food for Sustainability and Itacil, representing Portuguese and Spanish leadership respectively. Then we also have the National Institute of Agriculture and Veterinary Research from Portugal, representing academia. And we have the Union of Agricultural Cooperatives of Castilla and Leon, representing farmers. And of course, we have EIT Food representing industry and facilitating this international cooperation. Currently, we're working with regional authorities in both countries, namely Junta de Castilla y León and Comunidade Intermunicipal de Beira Baixa. We also have 10 farmers in total working with us already, 3 in Portugal and 7 in Spain. Currently, we're focusing on extensive crops, going into fruit orchards, herbal crops, vineyards, pastures, and livestock, often in relationship with agroforestry system traditional to our region, our cross-border region, commonly known as montado in Portuguese, or dehesa for our nuestros hermanos in the other side of the border. So, as I mentioned, our Living Lab is also is in the stage of preparation for the Living Lab launch event, which will take place next Tuesday, spoiler alert. And we have started engaging stakeholders already to be presented in our Living Lab. So we have sent out forms, emails, formal invitations. We've been interviewed by local newspapers, and we're trying to create as much as an awareness as we possibly can, and collecting data into stakeholders that want to collaborate with us. Right now, we've gathered over 39 manifests of interest from different stakeholders throughout our region, but not only. So going from Lisbon and Setúbal in center-south Portugal all the way to Valladolid in Spain. And most of them are actually farmers that are working in over 2,000 hectares of land. So in Portugal in particular, so far we're starting to engage 15 stakeholders coming from mostly academia and spread a bit all over the country, as I mentioned, not only from our region here in Beira Baixa. And most of the farmers that we have interviewed so far are working in organic farming and working with livestock as well. On the Spanish side, we'll have engaged 24 stakeholders so far, with most of them representing the most of the area that is actually being farmed in the Living Lab. The most representative class of the Spanish stakeholders are farmers that are working mostly with conventional agriculture, and the majority of them are not producing livestock. So a bit of a contrast to the Portuguese scenario so far. So we gathered all this data in our efforts to invite people to our Ibersoil launch event. This is the program that we have designed for it. As you see, we have 2 participative sessions being the focal point of our activities. We will ask participants to contribute with their thoughts into the way that the Living Lab should work and share their experiences with different carbon farming practices and what they think that carbon credit markets could be useful for their activities, including sharing their experiences with carbon credit schemes. It will also be a good day for networking, of course, with stakeholders coming mostly from Beira Baixa and Castilla and Leon. But we will also have MRV technology providers, researchers, regional authorities being represented. So we have a very diverse environment. And we will, of course, have a small tour around Ciudad Rodrigo where we will host this event. So I reach out to you if you or anyone you know is interested in participating in our Living Lab. Please feel free to attend our Living Lab launch event next Tuesday, the 25th of February in Ciudad Rodrigo, Spain. We will have free transportation from and to Idanganova, right here next to our headquarters. And please do take this opportunity to participate in this project from the very beginning by sending me an email. We don't have much time, but we still have some seats on the bus. So please, it's still not too late to come and join and start working with us. So if you want to also reach out, even though you cannot go to our event, please scan these QR codes, reach out to us, tell us what you're working at, what is in your interest, how do you want to collaborate with us, what do you need from us, yes, please feel free to reach out to us, and thank you very much for your attention and for sticking with us until this time. Thank you.
Thank you, David, and what a great opportunity you present here. Fantastic. So, Cristine, if you want just to leave your presentation open because the QR code is there. So, for our participants, Chris was just telling us how much money we can save from adopting this sustainable practice. Now you bring us the opportunity to implement some of this practices. You also have money to assist with presentation. So I don't think that we can ask more from this webinar. This is really a great opportunity. So fantastic. So let's get in touch with David if you want to know more and start to walk the path of the sustainable farming practice. Thank you so much, David. It was really interesting to hear this project, and best of luck. I'm very conscious about our time. I know that we have a hand raised, but we cannot allow hand raised. Apologies. So please write your question here at the chat. But just before we go, just two things, just to wrap up our webinar. So I know that some of you are really keen on having the certification. So please. Just fill in this very short questionnaire about our webinar here today, and we can then send you a certification for being here. And at the same time, I'm going to take the opportunity, since we still have Cristine and David and Noelia here, just to ask one final question, a roundup question. So we've been talking here about soil microbiome, soil health, these new opportunities. So from your perspective, how do you think that these advances in soil microbiome research might change our approach to sustainable agriculture and climate change mitigation in the coming years? So where do you think that our farming practices are heading with all this knowledge and opportunities, as David has brought us here, will lead us to changes in the future? Who would like to go? Cristine? You are head of strategy. Any thoughts on where this might lead us?
I think the most important thing that our knowledge on the soil microbiome can lead us is it just to underscore the importance of feeding that microbiome. And so that I think that's just going to lead to more scientific evidence that these practices that we call soil health management practices, climate smart practices, carbon farming practices, that they're all so vital to the agroecosystem. And I think it's just going to add to the mounting evidence that we have to feed the soils if we want them to perform at their best. So I don't think it's going to be a big revolution. There's not going to be a pill or certain amendment at the end of the day. I think it's hard work to practice good farming. It's not easy. And I think we just have to build the evidence that it is a worthwhile endeavor.
Yes, and with climate change and with the more European restrictions, it's also getting tougher to not to practice good farming systems. David, would you like to share some thoughts with us?
I fully agree with Cristine. I mean, we definitely need to start looking into these sorts of concepts as absolutely fundamental to have resilient food systems, especially in our region, in the Mediterranean area. I mean, we're looking into harsher and harsher climate. We need to sustain local food production. We need to give people the necessary income streams. We need to preserve all our landscape. So this is definitely a means to that very important end.
Yes, indeed. And as the world is turning, we also really need to be very careful about our food security. So we definitely need to care about that. It was great to have you here. And I also thank all the audience for staying, up to even past the hour of the webinar. It was really, really a pleasure. We have now come to an end of our first cycle of this year. But the team has already prepared the next cycle. So we will be back again on the 6th of March. This full cycle is dedicated to biostimulants. So if you are interested in the topic, we have prepared very interesting conversations again, just on biostimulants. I hope you can join. Please register. And in the meanwhile, we'll be making everything that we've discussed here in this past series available to you. They will be available on our website. So for me, it was a pleasure to be here conducting this session. A great, great thank you to our audience, and a special thank you also for our speakers. Okay, so see you on the 6th, if not before. Thank you.
Thank you all very much.